Welcome to Lecture 10 of BIOT 643. The lectures this week will cover molecular diagnostics and therapeutics. The outline for this lecture is, first, molecular diagnostics, direct detection, antigenic detection, nucleic acid detection, and this will include fluorescence in situ hybridization, or FISH. Molecular therapeutics, where we'll cover vaccines and gene therapy, and last, we'll be discussing stem cells. First, we'll start with molecular diagnostics. Molecular diagnostics is the ability to detect small biologic macromolecules or other molecules in soil, water, humans, animals, and plants. The desired features of molecular diagnostic tools are specificity with low or no false positive signals, sensitivity, the ability to detect the target organism at low concentrations from a crude mixture of multiple organisms, efficient, done in simple, easy steps, and cost-effective. The tools of molecular diagnostics have tremendous implications in the areas of oncology, monitoring disease progression of cancers, genetic diseases, identifying predisposition or early detection, drug testing, detection of metabolites, pathogens, and other infectious agents, tissue or organ transplantation, and targeted therapy. To use molecular diagnostics for detecting infectious diseases, there are basically four different possibilities for laboratory diagnosis of an infection. First, there is the direct detection of the pathogen, either by microscopy or by culture. In this lower diagram, this is shown by the upper right figure. There is the direct detection of nucleic acids of the pathogen, and this can be achieved by a number of methods, either the automated DNA sequencing, PCR, or detection of nucleic acids by DNA hybridization. Or there's the detection of the protein components of the pathogens with the use of specific antibodies. This would be an antigen capture ELISA. Here you have antibodies to specific components of a pathogen as your capture antibody. It captures antigens derived from that pathogen. The other option, number 4, is an IgA, IgM, or IgG specific detection of antibodies directed against a different pathogen or changes in their corresponding titer, and as a most sensitive method. For many infectious diseases, a patient will generate an antibody response against the pathogen, and one can detect the presence of those antibodies as a marker of the presence of disease. For many diseases such as hepatitis or HIV infections, one looks for the presence of antibodies against the hepatitis virus or the HIV virus, as opposed to looking for components of the virus itself. So let's talk about the direct detection. Well, the two main categories of direct detection are visual detection, which would be microscopic evaluation, or culture methods. This could be an in vitro culture or a mouse inoculation. For microscopic detection, the advantages are that it's simple. Direct detection of a pathogen. And their morphologically distinct organisms can be identified. The disadvantages are that it's slow and tedious. It requires a highly skilled technician, and in many cases cannot discriminate between highly similar organisms. The next detection method would be in vitro culturing or mouse inoculation. The advantages are that it detects only viable pathogens. It measures virulence and infectivity. Again, the disadvantages is that it's slow and tedious, and with the mouse inoculation, it uses animals, and with handling, a pathogen may lose its viability. So that leads us to antigenic detection. And for antigenic detections, antibodies are commonly used. An antibody is a protein or immunoglobulin that is produced by B cells and recognizes a specific site on an antigen, and this is called an epitope. An antigen is a molecule that induces the production of antibodies and has antigenic determinants or epitopes. The antigen-antibody interaction is the main principle utilized by immunologic methods used in molecular diagnostics. And here on the figures on the right, we show this is a diagram of an IgG molecule where you have the heavy chain and light chain. On the end of both the heavy chain and the light chain, there are what are called variable regions. These variable regions work together to form what is called the antigen binding site. And when you look at the next figure down, shows a graphic representation of an antigen. In this case, there are colored boxes attached to this antigen, and there are 7 of them. And this caricature represents that there are 7 different epitopes. Each one of these epitopes can elicit a different antibody. So each one of these antibodies, though it may react to the same protein, may react to different sites on the same protein. The advantages of antigen detection are that it's simple and fast. It can be automated. It has the potential for high throughput. The disadvantages are low specificity, and it does not always distinguish between latent and active infections. So what are the types of antibodies? There are polyclonal antibodies. These are raised against purified proteins. They are a mixture of antibodies directed against different epitopes of the same antigen. When exposed to an antigen, many different B cells are activated to produce antibodies. This results in a mixture of antibodies that could recognize different parts of an antigen. If you'll recall, in the previous figure we showed the model antigen which contains 7 different epitopes. So it is entirely possible that you will obtain 7 different antibodies, and this mixture would be called monoclonal or polyclonal antibodies. The main drawbacks to using these antibodies are the amounts of the different antibodies within a polyclonal preparation vary from one batch to the next. They will also vary between one animal and another that produce these antibodies. It can't be used to distinguish between 2 similar targets that differ in only one antigenic determinant. So polyclonal antibodies are used in the common immunologic technique termed ELISA, or enzyme-linked immunoabsorbent assay. This method involves sequential binding of the primary antibody that binds to the antigenic determinants, followed by the secondary antibody which binds the primary antibody. After each binding step, the unattached antibodies are removed by a wash step. Here we show this diagram on the right. First, you bind your antigen to the source, your support. So it's a solid. It can be a solid, say, polystyrene microtiter well. So your antigenic target binds to the support with the antigenic sites or epitopes exposed. You add your primary solution of polyclonal antibodies. These bind to the antigenic sites on the target molecule. Whatever antibodies don't bind are removed by washing. You then add a secondary antibody which is conjugated to an enzyme or a fluorophore. These bind to the primary antibody, so if your primary, the first antibody added in step B, is say derived from a rabbit, then the secondary antibody would be an anti-rabbit antibody that is conjugated. The secondary antibody binds to the primary antibody. Any unbound secondary antibody is then washed away. You now add a substrate and then determine the amount of this label that is bound to this in the secondary antibody that's bound to the primary antibody, which is proportional to the amount of antigen which is present. In contrast to polyclonal antibodies, there's a group of antibodies called monoclonal antibodies. Monoclonal antibodies are directed against a single antigenic determinant on an antigen. They are derived from a single type of B cell. In theory, they're actually derived from a single B cell. The use of monoclonal antibodies allows the identification of closely related antigens that differ only in one or two epitopes. These monoclonal antibody producing cells were initially produced in 1975. So how do you generate a monoclonal antibody? Well, there are 4 steps in the generation of monoclonal antibodies using hybridoma technology. First, there's the immunization. You have a mouse who is immunized with your target molecule of interest. The spleen cells are isolated from this immunized mouse. You extract the spleen, you mince and isolate the B cells. These B cells are then fused with the myeloma cell, which is an immortal B cell, is a form of B cell tumor that do not produce antibody chains. Commonly, a clone which is called NS1, which is a non-secretor cell, is the one which is most commonly used as a fusion partner for the production of monoclonal antibodies. In this diagram, we show the spleen cells isolated from the mouse that was immunized. They are fused with the myeloma cells. Then you have fusion partners. So once you have the fusion, these fused cells are selected by their ability to grow on what is called HAT media. This is hypoxanthine, aminopurine, and thymidine media. And the cells that produce a specific antibody to the immunized antigen are identified by immunoassay, followed by your antibody production, and the hybridoma is used as a source of these monoclonal antibodies. So this is very much like what we saw in the selection of vectors. These myeloma cells have the ability, neither the myeloma cells or the spleen cells can grow on HAT media, but the fusion partners can. So when these cells are grown in HAT media, only the fused cells are allowed to grow. You can then test, you take these fusion cells and you plate them in by what's called limiting dilution. That means you dilute them such that you apply 1 cell per microtiter well. Then you assess the media from that single cell to see if it is producing the antibody of interest. And that allows you to screen it and define a positive versus the negative producing clones. Because of the high specificity of monoclonal antibodies, they provide higher sensitivity when compared to polyclonal antibodies when used for molecular diagnostics. The targets for diagnostic monoclonal antibodies include tumor markers, polypeptide hormones, cytokines, drug monitoring, infectious diseases. The next topic we'll cover is nucleic acid detection. Nucleic acid-based methods are widely used in rapid and sensitive identification of the presence of pathogenic organisms or for the diagnosis of disease based on detection and analysis of genetic material. The detection of specific nucleic acids, and that can be either DNA or RNA, can be used to diagnose infections with viruses. Many diagnostic kits in use today are based on PCR analysis to determine the presence of a mutated gene or an infectious organism. Additional commercial uses of nucleic acid detection and identification include paternity testing and forensics. Also, many of these applications can work with alternative fluid specimens such as oral fluid, i.e. saliva, fluids from the eye, or urine. These nucleic acid-based diagnostic tests are directed toward a specific target sequence. They can be used to identify disease-causing organisms, such as bioterrorism agents. They can analyze forensic samples. They can differentiate plant genome patterns by rapid amplification of polymorphic DNA. They can identify single nucleotide polymorphisms, or SNPs. The key elements of a nucleic acid-based diagnostic system are: probe DNA should be highly specific. This enables the identification of the presence of the pathogenic organisms or the predisposition for a disease. For example, certain diseases result in genetic rearrangements at a defined loci. DNA probes can be used to identify these rearrangements. The hybridization probes can be labeled using either a radioactive or a non-radioactive molecule. The target DNA sequence can be used to design the DNA probes. And signal detection using fluorescent molecules provides a sensitive assay. For molecular beacons, these are the commonly used non-radioactive probes utilize a biotin-avidin or a biotin-streptavidin interaction. And they have the advantage of a much higher stability relative to radioactive probes and high sensitivity. An example of a non-radioactive probe is molecular beacons. Molecular beacons are typically 25 nucleotide long hairpin molecules with an internally quenched fluorophore whose fluorescence is restored when they bind to a target nucleic acid sequence. And here we show diagrammatically this type of image. Molecular beacons work similarly to a fluorophore-based quantitative PCR. Hybridization causes the separation of the stem, which in turn separates the fluorophore from the quencher. Illumination of the hybrid with light results in fluorescent emissions. And here we show a diagram of those activities on the right. Here, where you have your molecular beacon probe, it hybridizes to your target DNA probe, and instead of being in direct contact, your fluorophore and the quencher are now removed, separated from each other, allowing the fluorophore to fluoresce. So for these nucleic acid-based methods, there are a number of advantages. It's simple, fast, and specific. It can be automated. It has the potential for high throughput. It can distinguish different species. And it can detect parasite pathogens directly. It has certain disadvantages as well. It's expensive and it's multiple steps. It does not always distinguish between live and dead organisms, such that the possible false positives and false negatives. The next technique we'll talk about is fluorescence in situ hybridization, or FISH. So what is FISH? This is a technique that involves the hybridization of a fluorescently labeled nucleic acid probe to an immobilized target, such as a metaphase chromosome, an interphase nuclei, or tissue sections. In the diagrams at the bottom, we show photographs of, first, on the left, the whole chromosome has basically looking for chromosome 2 and 5. Using probes that are fluorescently labeled, one can detect those two different chromosomes. The interphase nuclei here uses a centromere probe for the ability to detect chromosomes within the interphase nuclei. And then the last figure shows the same approach in a tissue aspirate with target-specific probes. So for the general principle of FISH, probes can be directly or indirectly labeled. The denature probe and target the specimen. You add the probe to the target and allow it to hybridize. You wash to remove excess probe. You apply the fluorescent label, this is then the indirect condition only, and counterstain. Then there's the detection and imaging. So here we show on the right sort of a schematic of the direct and indirect labeling. With indirect direct labeling, a single stranded probe labeled with a non-fluorescent hapten. This is hybridized to single-stranded DNA from denatured chromosomes. Then you have a hybridization of the probe to the chromosome, then applying fluorescently labeled antibody against the hapten, and then visualization of the fluorescence using a fluorescent microscope. The second method is the direct labeling. Again, you have the single-stranded probe labeled with fluorescent deoxy-UTP. This is then hybridized to a single-stranded DNA from denatured chromosomes. The hybridization of the probe to the chromosomes allows the direct visualization using a fluorescent microscope. So what are the types of probes one can use? One can use repetitive probes, such as a centromeric probe, a telomere probes, a ribosomal DNA probe, or an interspersed repetitive elemental probes. Here are examples of these shown on the right. There are locus-specific probes, cDNAs, PCR products, cloned genomic DNAs. These can be bacteria artificial chromosomes or yeast artificial chromosomes. And finally, a type of probe are painted probes. This is a whole chromosome paint, an arm-specific paint, or a band-specific paint. And again, we show examples of each one of those are shown in the diagrams or photograph on the right. Then there are specialized forms of fish. In this case, we discuss the spectral karyotyping, or SKY, using a cocktail of all 24 human chromosomes differently labeled. This paints all chromosomes in the metaphase spread. This is useful in the analysis of complex karyotypes. Here on these lower images we show the spectral color image in a diploid, the spectral color image for an aneuploid, a DAPI banded image, and a classified image. The next topic are molecular therapeutics. Vaccines. A vaccine is an external agent that elicits an immunogenic response. That means it generates antibodies in an organism against a disease-causing organism. Traditional vaccines have been either against killed or inactivated organisms or live, non-virulent, or what they call attenuated organisms. Some of the newer vaccines may be one of the following. Subunit vaccines, where specific portions of a pathogenic organism can be used to generate a vaccine. The primary requirement is that the component should be able to elicit antibodies that react against the entire organisms. Some advantages of the subunit vaccines are well characterized, stable, safe, and free of exogenous proteins. Some examples include the coat protein from the herpes simplex virus and the hoof and mouth disease virus. There can be peptide vaccines. These are chemically synthesized peptides that mimic the epitopes. The major limitation is that the peptide's conformation should be similar to the native one, and the single epitope should be able to generate an immunogenic response. Then there are vector vaccines, naturally occurring viruses that infect the organism, i.e. humans, are modified to take the gene of interest that is infected into the organism, and the gene of interest is produced. The next are DNA vaccines. In this case, instead of introducing a protein, the gene encoding that protein of interest is introduced into the organism under the control of a eukaryotic promoter, where the protein is then expressed. The advantage of using DNA vaccines include low cost. The proteins are more likely to have the correct conformation because it is being produced inside the organism. Only the protein of interest is produced because it is the only one under the control of the eukaryotic transcriptional translational machinery. The next topic that we'll discuss are the molecular therapeutics, gene therapy. Each cell in the human body contains DNA, which is the individual's genetic blueprint. DNA is organized into genes, which in turn are organized into chromosomes. A change in the DNA sequence or code of a gene is called a mutation. Often, a mutation is harmless to the individual. However, some mutations can lead to serious diseases. To date, approximately 4,000 diseases have been attributed to genetic disorders. Gene therapy works by replacing a defective gene with a functional copy that will perform the normal functions and, in theory, fix the problems caused by the deficient gene. Gene therapy has the potential to treat a number of genetic and acquired diseases. It is performed with non-reproductive cells, generally known as somatic cells. It is possible to insert new genes into not only somatic cells, but also into more specialized reproductive cells, such that the corrected gene is passed on to successive generations. So the requirements for successful gene therapy treatment are the following: efficient transduction of the target gene, that is, delivery of the new gene into the target cell; long-term expression of the therapeutic gene, or how long is the gene expressed in the target cell; a lack of an immune response to the vector of the transduced cell; an absence of toxicity to the patient following delivery. So the critical component of gene therapy is the delivery of DNA into the host cell. There are various ways of introducing these genes into somatic cells. Two widely used methods of gene transfer are viral methods and non-viral methods. For viral methods, since viruses are naturally occurring organisms capable of transferring their genetic material into host cells, for use in gene therapy, the vector is most often a virus. The virus is engineered such that it has a low toxicity and high transfer potential. The design of a viral vector for use in gene therapy is illustrated below. Here we show the wild-type virus with its viral genome, and we place into this plasmid the viral structural genes. This is integrated into the viral genes and the viral structural proteins. This is how this normally occurs, but for a therapeutic gene, instead of the viral genome, this therapeutic gene is now inserted into the viral plasmid. This is now an integrated therapeutic gene, and this is made into the budded virus vectors. There has been significant progress made in the treatment of certain rare genetic diseases. However, fulfillment of the promise of gene therapy will require both improved gene delivery vehicles and the emergence of gene transfer systems that are compatible with standard medical practice. So for gene therapy delivery vehicles, the vectors commonly used as delivery vehicles include retroviral vectors, lentiviral vectors, adenoviral vectors, adeno-associated viral vectors, human herpes virus viral vectors, and pox virus. The use of retroviruses in gene therapy emerged in 1981, when several groups independently reported the development of replication defective and highly engineered recombinant murine-based retroviral vectors for the delivery of potentially therapeutic forms of wild-type genes to mammalian cells to correct the defects caused by a mutated gene. In 1983, a retroviral vector was used successfully for the first time to correct an enzymatic defect. Retroviral vectors have the following features that make them useful for gene therapy. The receptor for the envelope virus is ubiquitously expressed in mammalian cells. They integrate efficiently into the genome of the target cell. They do not transfer any viral gene, thus minimizing the risk of an immune response against the transduced cell. The genome and the life cycle of the retroviruses are relatively simple and well studied. This allows for continuous improvement in the vector design and generation of stable systems amenable to characterization and scale-up. They can transfer up to a 7 kilobyte piece of foreign DNA. The long terminal repeat, the LTR, of retroviruses contain an efficient promoter and enhancer that function in various cell types and can direct the expression of the gene inserted into the retroviral vector. However, there are some drawbacks to retroviral vectors. Despite their wide use, retroviral vectors suffer from one major drawback. It can only transduce dividing cells. Although retroviruses can enter non-dividing cells, the viral genome is excluded from the cell nucleus by the nuclear envelope, the breakdown of which occurs during the normal course of cell division. Therefore, the replication cycle of the retrovirus can be completed in an actively dividing cell. For example, hematopoietic stem cells, which rarely divide, terminally differentiated neutrons, and tumor cells, which do not all divide at the given moment. Another blow to the use of retroviral vectors in gene therapy came when two toddlers with fatal immune deficiency, the bubble boy disease, developed leukemia-like symptoms. In 2 out of the 10 children, the added gene inserted close to a gene known to play a role in leukemia and turned on that gene. One theory on why this occurred is that the retroviral vectors do not integrate as randomly as it is believed, and that increases the potential risk associated with these vectors. The use of retroviral vectors can therefore be limited in many potential clinical applications in which non-dividing cells are the targets for gene therapy. For lentiviral vectors, as compared with retroviruses, lentiviruses have a more complex genome and consequently are more complex replicative cycles. The HIV-1 is the most studied lentivirus. Lentiviral vector development is dependent upon the viral components that enable HIV to productively infect non-dividing and terminally differentiated cells. For example, macrophages and lymphocytes. However, because of the complex genome and replication cycle, it has been difficult to develop vectors and packaging lines of lentiviral origin. Adenoviral vectors. These are very efficient and transducing target cells. They possess properties advantageous for in vivo gene delivery of recombinant DNA into a wide variety of dividing and non-dividing cells. Because of their affinity for the respiratory epithelium, these vectors were first used to study gene transfer in patients with cystic fibrosis. The advantages to these adenoviral vectors are they can be grown to high titers or concentrations. They are more stable to concentration and purification. But they have disadvantages as well. They stimulate inflammatory and immune responses. They do not integrate into the host chromosome and thus the need for repetitive administration. Most often the second dose elicits an immune response. And they are only effective for short-term transgene expression. Here we show an example of the adenoviral vectors for gene therapy, where we show the adenovirus with the double-stranded DNA infecting a targeted cell, in this case the airway epithelial cell. And this is the intended outcome, in this case using the cystic fibrosis gene. The adeno-associated viral vectors can infect both dividing and non-dividing cells. These vectors have many natural features that are absent from alternative vectors, such as the lack of pathogenicity. They're stable in multiple cell types. They have a simple genome. They're easy to manipulate genetically. They usually target the central nervous system, the eye, and other sensory organs and skeletal muscles. Herpesviruses, viral vectors. These are derived from herpes viruses, which are large DNA viruses with the potential to accommodate large or multiple transgene cassettes. They can persist for lifelong in a latent state without causing disease in the immune-competent host. They have a broad host range and can infect non-dividing cells. Or poxvirus. These are double-stranded DNA viruses that can infect both dividing and non-dividing cells, capable of carrying large inserts, and these can be greater than 25 kilobases. They are being developed as vectors for cytokine gene delivery. They were used as vaccines for smallpox and have been shown to be able to carry multiple genes simultaneously. The use of these vectors is limited by toxicity of the vector, the potential inflammatory immune response, and the difficulty in targeting of the recombinant vector to specific cell populations. Also, there are non-viral methods for using transgenes, such as naked DNA. In 1989, John Wolfe, a gene therapist at the University of Wisconsin-Madison, made the discovery that naked DNA, when injected into mice, could be expressed. In 1994, Jeffrey Eisner and colleagues successfully injected an intra-arterial injection of naked DNA encoding vascular endothelial growth factor, or VGEF, to promote sprouting of new blood vessels. In 1998, a protein from a malaria parasite, when injected into human cells on a plasmid, provoked a strong immune response. And then in early 2001, Harriet Robinson of Emory University and other colleagues reported that a naked DNA vaccine could confer immunity in a monkey model of AIDS. Then there's lipofection. Naked DNA injections have only worked well in liver and muscles. To introduce genes into other tissues, lipofection is one of the techniques commonly used. A liposome is a microscopic sphere containing fat molecules. Coating the DNA, the liposome is absorbed through the fat-based cell membrane into the cell cytoplasm, where the genes are released. Modifications include various combinations of lipids and polymers. Researchers have developed a lipid-coated plasmid that is injected directly into tumors to deliver a gene that evokes an immune response against the tumor. In phase 2 trials, the immune system shrank the tumors and prolonged life in 8 out of 73 patients with advanced melanoma who failed to respond to other treatments. Electroporation. This method is commonly used to transfer DNA into bacterial cells. has also been used in gene therapy. DNA is injected into skin tissue, or the target tissue, which could be skin, muscle, or tumors, and uses a specially designed electrode to apply an electric field which punches temporary holes in the cell membrane that allows DNA into the cell. Lou Smith recently used this technique to transfer a blood clotting gene to hemophiliac dogs, temporarily eliminating the symptoms. Richard Heller's team at the University of South Florida were successful in delivering a cancer-fighting gene, interleukin 12, into skin tumors, causing some of them to disappear in mice. Other viral methods include a gene gun. These are DNA-coated gold particles are fired into the cell wall with helium pressurized gas. This is similar to what we discussed in plant transformation. Another method is microinjection. This is used for delivery of recombinant DNA into cultured somatic cells using microcapillaries. The major disadvantage of this technique is the limitation in the number of cells that can be injected at a certain time. The following are some of the techniques for gene delivery into human and mammalian cells that are currently in the developmental stages. The first is the triplex forming oligonucleotide. These are short, single-stranded DNA strands recognizing the double-stranded DNA with very similar or identical sequences, and nestle themselves into the double helix to form a triple helix. This method is used both to correct mutations in damaged genes and to introduce mutations to stop the production of defective proteins. There is small fragment homologous replacement. This works on the principle of homologous recombination and the cell's ability to exchange pieces of DNA between chromosomes. The next is the ex vivo approach. Cells are removed from the body and genes are inserted into them, and the modified cells propagate in the laboratory before being injected into the abdominal cavity. This method was used to transfer a gene encoding the blood clotting factor into skin cells. When the cells return to the body, they produce the clotting protein. The last group that we'll discuss are stem cells. These are the most controversial aspect of gene therapy. Humans, like some other organisms, have the ability to regenerate certain tissues, including skin, blood, bone, etc. The cells that allow for these tissues to be regenerated are called stem cells. These were first identified in bone marrow. This discovery, first made in the 1950s, created a lot of excitement in the field of regenerative medicine. In 1998, researchers from the University of Wisconsin-Madison for the first time isolated the human embryonic stem cells and kept them alive in the laboratory. There are over 200 different cell types in the body, and they all come from a pool of stem cells in the embryo. These embryonic stem cells are unique because they have the ability to develop into the 200 different types of cells in the body, or in other words, they are pluripotent. So, what is the controversy over stem cells? Embryonic stem cells are derived from the extra blastocysts that would have otherwise been discarded following IVF. Extracting stem cells destroys the developing embryo. So this raises certain questions, such as, is an embryo a human? Is it morally acceptable to use embryos for research? And when do we become humans? But in terms of pluripotent stem cells, here we show diagrammatically that pluripotent stem cells have the potential to become any type of cell. Here we show that the original stem cell is isolated from the cells in the inner mass of the blastocyst. These stem cells are placed in culture where they are grown under specific conditions which allow them to differentiate into pancreatic islet cells, hematopoietic cells, cardiomyocytes, neurons, hepatocytes. There are three important features of all stem cells. They can divide and propagate for long periods of time. They are undifferentiated. They have the ability to differentiate into a specific cell type. In terms of the types of embryonic stem cells, there are really three. First, there is the totipotent stem cells. These are derived following the fertilization of the egg at the 8-cell stage. This cell can form both the embryo and the placenta and all resulting tissues. There is the pluripotent cell line. This is derived from one of the cells within the intermass of the blastocyst. This cell can only form the embryo and cells derived from the embryo. And lastly, there are the multipotent cells. These are derived from tissue-specific stem cells, and they can only be deriving into tissues of the same tissue type. So here is a little bit better description. The totipotent cells, each cell can develop into a new individual. These are cells from early embryos, days 1 through 3. The pluripotent stem cells, these are cells that can form any cell types of over 200. These are cells that are derived from the inner mass of the blastocyst at days 5 to 14. Then there's the multipotent stem cells. These are cells that are differentiated but can still form a number of other tissues. These can be derived from fetal tissue, cord blood, and adult stem cells. Adult stem cells are those stem cells which typically give rise to the types of cells that reside in a particular tissue from which they are derived. For example, a blood-forming or hematopoietic adult stem cell derived from the bone marrow will give rise to different types of blood-forming cells. Experiments over the last few years have been successful in exploring the concept of plasticity, in which cells from one type of tissue can be directed to differentiate into cells of another type of tissue. Now, embryonic stem cells, these can be generated in really three different ways. One, following in vitro fertilization, derived from the intercell mass of the blastocyst, which is the stage of embryos at 5 days after fertilization. This intercell mass is made of 30 to 40 cells that are pluripotent in nature. For research purposes, these cells are removed and grown in a nutrient-rich media in a culture dish. Because of the ways these cells are derived, this research raises certain ethical questions. Second method is nuclear transfer. The nucleus from an already differentiated adult cell is transferred into an egg that has had its nucleus removed. The egg now contains the genetic material of the differentiated adult cell and is induced to form a blastocyst from which the intercell mass containing the embryonic stem cells can be retrieved. And third is the reprogramming of adult stem cells. This was introduced as an alternative to using embryos, where an adult cell will be made to act like an embryonic stem cell. As cells become specialized, they in turn lose the ability to turn on genes required for the versatility offered by embryonic stem cells. This silencing of these genes is believed to make cells specialized and lose the differentiating capacity. More recently, a source of stem cells has been the umbilical cord stem cells. These are adult stem cells of infant origin. They are less invasive than bone marrow stem cells. They have greater capabilities and they are less expensive. So why stem cells? What is the utility of using them? Well, very few diseases can currently be treated because scientists can only treat or generate a few types of tissues, most successful of which is blood and skin. Research in stem cells may provide a cure for diseases such as type 1 diabetes, where stem cells can be stimulated to produce insulin-producing cells, or in Parkinson's disease, where certain types of nerve cells are missing. For example, in type 1 diabetes, pancreatic cells do not produce insulin. Embryonic stem cells might be trained to become pancreatic islet cells where they can produce and secrete insulin. In heart disease, adult bone marrow stem cells can be injected into the hearts and are believed to improve cardiac function in victims of heart failure and heart attacks. The new areas of research are reprogramming of cells. And here we show an example of creating induced pluripotent stem cells. First, you isolate cells from a patient, either skin or fibroblast, and you grow these on a dish. You treat these cells with reprogramming factors. You then wait several weeks under these culturing conditions. At that point, you now have differentiated into pluripotent stem cells. You can change the culture conditions in order to stimulate these cells to differentiate into a variety of cell types, from blood cells, like cells from the gut, and cardiac muscle cells. So why are stem cell research so important to all of us? Stem cells can replace diseased or damaged cells. Stem cells allow us to study development and genetics. And stem cells can be used to test different substances, drugs, and chemicals.